ser en inglés, él entiende unas pocas palabritas en español, y hacia el final, pues dejamos un espacio para preguntas. Entonces, Colin, welcome to the seminar. Thank you for accepting the invitation. I already present you what you are doing right now as a PhD student, and already explained a little bit on the reasons why we pick up this theme of your master thesis to join this seminar today. So we're all ears, and please go on with your presentation.
Thank you, Dr. Mateo Lopez Victoria. I appreciate the introduction. And let me see if I can, this will stop sharing my screen for the presentation. Okay, can everybody see that? All right. Again, thank you so much, Dr. Mateo Lopez Victoria, for the introduction, and greetings, everybody. My name is Colin Howe, and I am a current PhD student at Penn State University, working with Dr. Monica Medina, who's also originally from Cali. As Dr. Mateo said earlier, I am here in Cali right now, collaborating with the Universidad de Javeriana de Cali, and so I'm very gracious and I'm excited to share a little bit about my research that I did for my master's thesis back in 2015. And the title of my talk today is The Acclimatization of the Fused Staghorn Coral, a Cropper Prolifera to Non-Native Locations within the U.S. Virgin Islands. Now, to start off really quickly, the overall goal of my master's research was to develop a baseline health assessment for Cropper prolifera within a coral restoration framework. And in today's presentation, I would like to discuss a little bit about why this is important. I'll talk a little bit about the history of Acropora corals in the Caribbean for those of you who may not be familiar with the Acropora genus. Talk a little bit about coral restoration and how that role has expanded in the effort to try to restore and to repopulate coral reefs in the Caribbean, and what interesting and exciting questions and what opportunities researchers have in using Acropora prolifera within these methodologies. So taking a step back, historically, for many of us will know that two species, Acropora silvicornis and Acropora palmata, have survived and basically covered the entire Caribbean basin for hundreds of thousands of millions of years. And they have a few interesting characteristics: rapid growth rates, up to 10 centimeters per year. But then they also have a really interesting ability of high rates of asexual reproduction due to fragmentation. So branches of these two colonies, of these two species, can break apart, will reattach themselves to the substrate, and then continue to grow colonies. And this life history trait creates large thickets, huge, wide, expansive ranges of coral cover. And this kind of colonization provides a lot of very important and irreplaceable ecosystem services. One, these services that provide is dense habitat. Because of this three-dimensional branching morphology, it creates a lot of habitat that countless and a large number of diverse marine organisms can live and utilize. And that's extremely important in maintaining the resiliency and the functionality of coral reef ecosystems. But then these types of corals also provide other ecosystem services in terms of economics, in terms of providing resources for us to eat for a lot of the fish that they'll provide habitat to. That's also a benefit for us. But then these are actually beautiful shallow water corals, and a lot of tourists love to see these types of coral reefs. But then another ecosystem service is the coastal protection they provide. Again, with that branching morphology, it does a very good job of dissipating energy from offshore waves, and it can protect coastline communities, people who have their homes along the coast. And
Quiero preguntar si de pronto yo soy el único al que se le paralizó la pantalla. Ok, voy a comunicarme con Colin a ver qué le pasó. Creo que se desconectó al internet. Denme por favor un minuto. Colin me acaba de decir que se quedó sin internet. Él está aquí en Cali como parte de su plan de trabajo con nosotros en la universidad. Se está quedando en un Airbnb ahí en el oeste. Aparentemente se fue la señal del internet. Está tratando de averiguar si es algo del router en la casa donde está, en el apartamento donde está, o es un problema, digamos, más serio. Entonces, denme un minutico, yo me comunico con él a ver qué solución le podemos dar.
Lo
Y ya,
siento, everybody.
ya volvió.
All right, let me see if I can reshare my screen again. My
Oh, no
internet
problem.
has been giving me problems.
Okay. We're still
Okay,
here.
perfect. You guys can see this?
Yeah, go
All
ahead.
righty. Wonderful. So I was talking about the population decline of both of these two species due to a number of compounding stressors. And there were hurricanes, bleaching events, cold fronts, but the most severe cause for this decline was an outbreak of white band disease. And I mentioned earlier about this high rates of asexual fragmentation. And so, for example, if you take a look at the photos on your right, some of these thickets of large, dense populations of colonies could only contain one or few genetic genotypes or unique individuals in terms of their genetic traits. And so this disease had a profound, a very severe effect on these populations, which led to the large decline in the abundance and cover. And with that, all of those vital ecosystem services that we highly depend on with these corals were also lost. And this was a very serious situation. These two species were actually included into the ICUN Red Endangered Species List in 2006, one of the first coral species to be included onto this list. And because of that, a lot of interest started to shift into how can we repopulate these corals, and thus it actually facilitated and kind of spurred the development of coral restoration efforts and methodologies in the Caribbean. Coral restoration has actually been around for a long time. It's gotten its start in the Pacific and other areas. However, just as this decline started to occur, we started to see a huge push for coral restoration efforts in the Caribbean. Now, in short, this is a very condensed definition or goal of coral restoration. It's such a dynamic new field. There are a lot of different objectives and methodologies depending on what marine practitioners and marine managers are trying to accomplish. But overall, the goal of coral restoration is to reestablish the functionality and the resiliency of coral reef ecosystems to where these ecosystems can persist naturally on their own. And so reestablishing coral populations and density, bringing back that dense habitat and structure, inviting those marine organisms to occupy those space, and for those systems to be resilient in light of future environmental conditions like climate change. Now, there are a lot of different methodologies that can go into it, but there are three main components. If you look at the photographs on the bottom, they are actually each step of the coral restoration process. The first process is to collect fragments from wild colonies, but they want to target multiple species. They are indeed primarily focused on the Acropora genus. However, there are certain restoration practitioners in the Caribbean who focus on Orbicellas. They focus on Montastria species. They even look at Millipora, we call it fire coral, because it does provide some of that structure. And so they're targeting multiple species, and they're also targeting multiple genotypes of these species, so multiple individuals to try to increase the genetic diversity. The next step is to include them in some kind of underwater structure. And there are a lot of different structures. One of the most popular and well-known is a coral tree that you can see in the center photo in the middle. You can hang corals up on these trees. These trees are suspended into the water column with floats on the top. And these trees do a number of provide a lot of benefits to these corals. It excludes a lot of the predators like the corallivore snails and the Hermodice fireworms. It also reduces the amount of sedimentation and competition from algae and other corals out on the substrate. It increases the availability for light and actually more flow sitting up into the water column. And it also allows corals a 360-degree plane for growth. And so in many studies, you will see even higher growth rates, especially for the Acropora corals in these tree structures. And then after about a year, these corals will start from about 5 to 10 centimeters and get to about a basketball or football size colony. And then you can fragment those again. And in the final photo, reattach them into the substrate. Now, identifying the locations ideal for coral restoration is also important. If you can see in this photo, this is a great example because there are critical species like Diadema antillarium, the long spine sea urchin, and other healthy corals. And also other aspects for why we target certain locations are if we've seen evidence of Acropora corals surviving there in the past. And so there are a lot of different methods and metrics that go into identifying where we can outplant and study these corals. Now, back in 2015, when I started this research, there were a lot of questions that we still did not fully understand in regards to the hybrid coral, Acropora prolifera. And a lot of these questions are very important that need to be answered first before we can suggest that we continue to propagate this hybrid coral. How long has this coral taxa been present in the Caribbean ecosystem? A lot of questions in the early 50s and 60s where they believed it was a unique morphology of either Cervicornis or Palmata, but not a hybrid in itself. But in fact, this hybrid coral has existed thanks to the geological records for over 100,000 years. The photo on the top left, on the top right of the screen is actually my photo taken from the Museum of Natural History in Washington, D.C. at the Smithsonian, where they've collected prolifera colonies dating back to the late 1890s, so before the 1900s. And prolifera was actually identified in 1816. So prolifera does have a historical role in the Caribbean. And then if you fast forward a couple hundred years up until the early 2000s, thanks to the advancement of technology in molecular studies, the prolifera hybrid was identified that it was indeed a hybrid using molecular DNA analysis in 2000. And then additional studies using these techniques, I also identified that evidence of introgression. Now, introgression is a fancy scientific term where they identify certain alleles from another species into another species. And it was suggested that because prolifera is a combination of genetic alleles from both species, from Palmata and from Cervicornis, that prolifera, if it could reproduce, would actually introgress those genes into the colonies that it reproduced with. And indeed, they did identify some Cervicornis colonies that contained alleles that were found within the Palmata genetic traits. And so that was one of the earliest evidences of this hybrid being able to reproduce. Another big question about hybrid animals in general are, with this combination of genetic information from two different species, there may be some deleterious genes or traits or genetic incompatibilities that limit its ability to survive in the wild. One of the most easiest examples is reproduction. However, Prolifera, this hybrid coral, can in fact reproduce, and actual visual observations of this were conducted in 2006 during spawning events in Belize, and then again in 2015. Now, what's really interesting, and I think this is because we haven't fully, this hybrid taxa isn't fully studied all that well. We have yet to find what is considered an F2 hybrid. And so that is two Prolifera colonies reproducing together to produce an offspring. But hopefully, as we continue to do more research, we'll find this, this elusive colony. And so we've answered a lot of questions about the history of the hybrid in the Caribbean. It's not a unique morphology. They've confirmed that it is indeed a hybrid. And so far, it does reproduce, and so there hasn't been any real signs of reduced by our fitness. The next questions that we needed to answer before thinking about using them in coral restoration was the ecological viability of this coral hybrid. Again, do we see any genetic traits or any deleterious genes that limit its ability to survive in the wild? What kind of stressors may, how do this coral respond to certain stressors that are related to coral restoration methods and relocation to novel locations, and how will this respond? And so these were the next questions that I aim to look at. Unfortunately, there's not a lot of great papers. There's only a handful of studies published on looking at Prolifera within an ecological framework. This paper that was done by Dr. Austin Bowden Kirby in 2008 did a reciprocal transplant experiment where he took fragments of Cervicornis and of Palmata and used what is called A-frames, so they're metal bars, and took colonies from the back reef and moved them to the fore reef and vice versa. And he compared their growth rates. Growth rates are one of the best ways to monitor coral reef health or coral health. And in this paper, he identified that growth rates do vary across genotypes. So unique individuals have higher or lower growth rates, so there's variation between that. And he also concluded that site adaptation, so where these colonies come from, actually influence their abilities to survive in a new location. One of my committee members, Dr. Nicole Fogarty, who has done a lot of research on Prolifera, she's one of the more seminal figures on this, this hybrid taxa, did an ecological comparison to both Palmata and Cervicornis in Belize. And from this graph, I hope you to bring your attention to, she did a linear transects and calculated the proportions of colonies surveyed that had different afflictions, disease, signs of parasitism, if they were bleaching or paling, if there were signs of predation or interaction with macroalgae. And from this, what I would to explain is that compared to the parental species, Prolifera is equally viable. And so what Dr. Fogarty and her research team found was that this hybrid taxa can survive in the environment equally to the parental species. Some situations it actually performed better, some it performed worse, like the macroalgae issue. And so it's not, this hybrid seems to be robust, but maybe not so robust over the parental species. And so with all of this previous information and research at hand, with no signs of genetic incompatibilities, it was historically present, these colonies are reproductively viable, and the Prolifera is equally viable to the parental species, we could finally argue that Prolifera could be, we can look at Prolifera within a coral restoration framework. And so for my research, I was really interested in understanding the physiological responses of Prolifera within this framework. For example, how does Prolifera respond to being taken from a colony, taken up out of the water and onto a boat, driven around the entire island, put into a completely new location, and how does that survive compared to the natal population of Prolifera in that particular location? Do we see similar variations across genotypes, so unique genets of Acropora prolifera? And what are some of the physiological and environmental, and I was planning to use some of the physiological and environmental metrics for health, like temperature, growth rates, and afflictions that Dr. Nicole, I mentioned earlier in Dr. Nicole Fogarty's paper, predation, mortality, bleaching, and disease. So I conducted my study site in the US Virgin Islands. You can see here the northern site. This is Introgression. This sits in the Atlantic Ocean here. And Flat K is the southern site. This is in the Caribbean Ocean. And for context, right here is the University of the Virgin Islands where I did my master's. And so it was a very quick trip that I could go out monthly to monitor my corals, which made it a lot easier. And what I did was a very simple common garden experiment. I went to northern Introgression and identified 8 individual colonies of prolifera that were at least 10 meters apart so that I could try to capture as much genetic variation as possible. And then I took them up out of the water. I took 8 fragments per colony. I'm sorry, I took 10 colonies or 10 fragments per colony, took them up out of the water and put them in a nursery for 2 weeks, and then outplanted them in the Flat K location. And I did the same thing for colonies at Flat K as well. So Flat K would be the control group, and the Introgression colonies would be the transplantation group. At Flat K, I attached 40 fragments in 2 1x2 meter plots. And part of the restoration methodologies calls for direct application to the substrate. So I use underwater epoxy and attach these corals directly to the substrate. I labeled each and every single colony with a tag so I can know where it came from and what colony it came from and which location. And I also included hobo temperature loggers that recorded temperature consistently. And I maintained the size of each fragment between 5 to 8 centimeters in length with 1 to 3 apical tips. For the experimental design, I decided to monitor these corals across 3 different growth rate axes. So vertical from the top, from the bottom, horizontal from either side, and perpendicular, basically the width of the base of the colony itself. Because prolifera has this unique branching morphology, it's very difficult to use a common technique, which is called total linear extension. And so to try to capture as much growth as possible, I decided to go with these 3 different growth rate metrics. I monitored each of these corals every month for 9 months, and I was able to calculate growth rate as centimeters per month. But in terms of growth rate, I also wanted to look at afflictions, what I mentioned before. And I monitored these in terms of presence, absence for mortality, bleaching, predation, and disease. For the statistics, I use what is called a linear mixed effects model, which is a great way to do everything all in once, where I used growth rates were compared, and the location was the main effect. So that calculated for the main effect of the differences between these corals. But then there was a mixed effect or a nested effect that I wanted to look at the genotypes. So how, were there any significant differences across genotypes? And linear mixed effects models allow me to do both in one test. And for the afflictions, I decided to use a generalized linear mixed effects model with a binomial distribution. My hypotheses were simple. I hypothesized that the natal population, so the corals that I collected from Flat K that were clipped from their colonies and reattached into the same location, would have significantly higher growth rates compared to the transplanted group. And for the second hypothesis, I also hypothesized that the natal population would have fewer signs of disease, predation, mortality, and bleaching compared to the transplanted group. So I assumed that the transplanted group would have less physiological, or would be more stressed and show signs of that stress. This is just the results from the microsatellite comparison for the unique genotypes. This was developed by Dr. Ileana Bombs at Penn State University. And the analysis was actually done thanks to my committee member, Dr. Nicole Fogarty at Nova Southeastern University. And this is a busy graph, but what I want to bring your attention to are the genet column, where you can see we identified at least 3 unique genotypes of prolifera colonies from both locations. However, some of these genets are actually still or all of the same colony, and so there is still high rates of asexual fragmentation at both of these locations. In a nutshell, based on all of the different metrics that I studied in terms of growth rates, we found no significant differences in growth rate from the natal population. So fragments from flat K, you can see the ones in orange are from the natal population, so they were the control, compared to the transplanted group, which is in blue, from interbrass. And so therefore, I failed to reject my null hypothesis for growth, and that I can confirm that transplantation stress does not limit growth for the transplanted colonies. Here, most growth rate studies provide growth, long-term growth results. And so here's just a quick snapshot, and these are combined just from the two sites from interbrass, from flat K on the top and interbrass on the bottom. Now, in terms of afflictions, we did see some significant results. Mortality and bleaching were not significant. However, we saw significantly higher predation rates on the transplanted group compared to the natal population. And what's even more interesting was that we detected genet variation as well. So within the genets of corals from interbrass, two of the genotypes showed significantly higher rates of predation compared to the third, which showed none, which was really interesting, interesting finding. Now, in terms of predation, this is more of an observational conclusion, but I wanted to bring your attention to apical tips growth rate. So additional growth rate that I monitored was the number of apical tips that these corals would produce. All acropora corals in the Caribbean grow apically. The white tips on each of the ends of those corals are actually the most productive part of the colony where a lot of the growth occurs. And I noticed early in the studies that Prolifera tends to put out a lot of apical tips very early in the growth. And I was able to quantify that. And so over the 9 months, they basically put out 9 to 10 apical tips in total, which was really high. And this actually has a benefit, I believe. Here's a great example of a colony. This is from the natal population from flat K. Started off with only 3 of these apical tips, and then within 9 short months created 18 of them. And now this morphology can actually provide an ecological benefit in terms of predation. I don't know how many of you have ever seen in the wild a fireworm or the Hermitaceae fireworm. That's what we call it in the States. And this is a corallivore. And this is about 15 to 18 centimeters. So it's a pretty big fireworm. And as we were doing my research, we saw it crawling straight through my site, but did not attach or did not choose to eat any of my Prolifera colonies. And instead, it found some small Acropora cervicarnis fragments. And if you can see how this worm devours the coral, it actually engulfs its body over the top of the colony, releases digestive enzymes and digests the live tissue, and then slowly pulls itself back up. And you can see a great example of this here. This fireworm had a big feast on this colony, and he's now going for seconds. Now, if I go back to this original photo of this Prolifera colony, this high apical tip formation, this intermediate branching morphology, actually may provide a deterrence or a difficulty for this type of predation. Now, the predation that I saw in my research was from a small snail. It's called Coralliphora, and it loves to eat, and it was mainly around the base of these colonies. And so this morphology does not exclude all types of predation, but in certain circumstances, I do believe that it can reduce predation, especially from fireworms, big fireworms like this. So in conclusion, overall, we observed no reduced fitness of the transplanted fragments compared to the control group. A. prolifera can survive outside of their natal habitat, which is really important if we want to try to incorporate this within coral restoration methodologies. And in fact, although this was not significant, the transplanted group that came from the northern side of St. Thomas actually more energetic in terms of wave energy and current and flow. And the transplanted group actually had higher growth rates, not significant compared to the natal population. And if we think back to the earlier paper by Dr. Austin Bowden Kirby, this also confirms the idea that site location may have a huge impact on the viability of these fragments. And we're seeing the same thing for the hybrid coral. Now, it is important to take into consideration within coral restoration methodologies, if you're targeting a site and you want to try to incorporate prolifera, based on our results, it's important to look at the density of predators at these locations. We saw that predation had a significant effect on the transplanted group. And so if you're transplanting fragments into a new location, taking into consideration the amount of predation that's at this site would be important. And we also did mention earlier, I mentioned a little earlier that there's a potential morphological advantage in the early stages of outplanting for prolifera with these high production of apical tips. Now, my study was done back in 2015, and since then, there's been a lot of new research and results that have come out. A graduate student from Nova Southeastern University, her name is Casey, did a similar experiment with prolifera within a coral restoration framework in coral trees. She suspended the prolifera fragments in the coral trees to monitor their growth rates compared to the parental species. And what she was able to find is that prolifera does in fact have higher growth rates compared to the parental species within her coral nursery tree. Although it's important to mention that these trees were in shallow waters between 3 and 2 meters of depth. There are also nonprofit examples of high success with Acropora prolifera. These photos are taken from a nonprofit called Fragments of Hope that's in Belize, run by a woman named Lisa Karn. And she and her team has done a phenomenal job restoring wild populations of all 3 Acroporas, but her team has found a lot of success incorporating prolifera within their coral restoration methodologies. And what's even more interesting, starting in 2010, we started to see population increases of Acropora prolifera in certain pockets throughout the Caribbean, the dry tortugas in the United States, Belize, Guadeloupe, Mexico, Puerto Rico, Jamaica, and in the US Virgin Islands. My professor, Dr. Nicole Fogarty, did some research in Belize and was studying the diversity of some of these populations and identified up to 17 distinct genotypes in certain locations. And what that suggests is that there are high rates of sexual reproduction between 2 species, Cervicornis and Palmata, that are generating these unique genotypes, because we still have yet to find the F2 hybrid. These are not prolifera reproducing together. They are Cervicornis and Palmata, multiple species, multiple genets of these 2 species reproducing together. And so something was really interesting. Dr. Fogarty came up with kind of an explanation for why we might be seeing this today. But firstly, this is photos from the US Virgin Islands. It's one of the study sites where I was able to collect some of my fragments. And something that's very interesting to note, this location right here, this is a place called Sapphire that's on the eastern side, closer to St. John in the British Virgin Islands. And Dr. Fogarty came to take some genetic samples from here. And apparently this is all one genet, one genotype of prolifera. So this is one colony that grew, fragmented. Those colonies grew, fragmented again, and produced this entire thicket of colonies. Now, this is also a great example of a monoculture. And this area is in risk if disease comes at this location. This might actually also be reduced. However, prolifera does seem to be surviving in present-day environmental and ecological conditions, despite the pressures of climate change. Now, what Dr. Fogarty hypothesized is why we're seeing high densities of Prolifera and why we're also seeing high genetic diversity within these populations is something she called the density-dependent reproductive isolation theory. And it goes something similar to this. Historic abundance of these two populations were very high, right? And both of these two species reproduce in a similar time frames. And when they reproduce, their gametes float up into the water column and then they mix. During the historic population, where there were high densities of both of these species, the likelihood of hybridization was rare because the distance between two reproductive colonies was so close that they were more likely to reproduce with one another than reproduce with a unique species. However, into present day environmental conditions, this is not the case. We're seeing such a reduction, and so the distance between two reproductive colonies is increased. Therefore, when these colonies reproduce and release their gametes into the water column, the likelihood that they will reproduce with a different species is higher. And so what she theorized is that there may be a mechanism geared towards hybridization, and why we may be seeing more Acropora prolifera in the near future. Now, when thinking about incorporating a robust, resilient hybrid into coral restoration methodologies, especially when we're also trying to reestablish the parental species, there are certain important things that we need to take into consideration. One potential risk from having a robust reproductive hybrid is something called outbreeding. And this is if the hybrid coral is so robust that it outperforms the parental species. This could overall decrease species diversity over evolutionary timescales, which is something we do not want. We want to increase species diversity. Or another issue would be inbreeding. Because Prolifera, it can reproduce back with both parental species. I forgot to mention that earlier. Prolifera has been shown to reproduce with both Cervicornis and Palmata. However, Cervicornis eggs are more willing to accept sperm from heterospecifics, so it can accept sperm from Palmata and from Prolifera readily. However, Palmata eggs are a little bit more, there are a little bit more prezygotic barriers that limit that. However, it can go both ways. Now, if these rates are high and Prolifera can reproduce back with both parental species at a high frequency, it would kind of dull the genetic distinction. It would limit the genetic distinction between species and overall could reduce genetic diversity, which would potentially bring these species closer to another catastrophic event if an outbreak of disease affected these colonies. And so there are certain things that we need to take into consideration. And when coral restoration practitioners are deciding on whether they want to use Prolifera, having an understanding of the genetic diversity of their targeted locations is very important. However, it's also important to mention that based on what we know so far and from previous studies, Prolifera seems to occupy its own niche. It actually occupies an intermediate habitat between Palmata, which prefers a shallower water, 1 to 2 meters, compared to the Cervicornis, which can survive in deeper waters. I've seen Cervicornis down at 26 and 27 meters. They're huge, thick colonies like that. They're amazing. And there was a previous graduate student at my university in the U.S. Virgin Islands who studied Prolifera across different depth ranges and identified that Prolifera does better in shallow water environments, but in deeper water, it cannot outcompete the parental species. And so therefore, this hybrid is robust, but the outbreeding potential is low. Also, overall, throughout the entire Caribbean range, there's low density of all 3 acroporids, and therefore there's overall low sexual reproduction to begin with. And so the risk of inbreeding from this hybrid coral to the parental species and ultimately the gene flow and introgression is also low. Interestingly enough, there was a study that was conducted in Puerto Rico that identified that introgression is actually a good thing that can benefit degraded populations like Cervicornis. they identified that in certain locations within the cervicornis in Puerto Rico, thanks to the back crossing, so thanks to Prolifera reproducing back of cervicornis, it actually increased the genetic diversity within the cervicornis populations. And this is not a unique idea or thought process that has been addressed when thinking about this hybrid coral. There was a really great paper by Dr. Willis et al. In 2006, where they identified that Prolifera represents an important reservoir of novel genetic diversity that may facilitate adaptive radiation under changes in environmental circumstances. Their ability to pass between genes between species represents a potentially significant mechanism for rapid evolutionary change of the parental species. And what this paper was arguing is that because Prolifera is a combination of unique genetic traits that contain unique traits that may allow its ability to survive in the wild, and because we're also seeing currently increases of Prolifera despite increased temperature and despite increased pollution, we're still seeing increases of Prolifera. They seem to be responding or to recovering quicker than the parental species. They have the potential to transfer these beneficial traits back to the parental species, and ultimately we may be able to increase genetic diversity if we incorporate Prolifera within coral restoration methodologies in a proper and controlled manner. And so for future considerations, just like any great scientist, when you've completed a study and you've done all your reading, you always are left with more questions. And so we want to look into the potential for incorporating a crop of Prolifera within coral restoration methodologies. So far, based on my results, we can successfully do this. And I argue that it is something that coral restoration practitioners need to do if they want to accomplish those goals of increasing resiliency and functionality within coral reef ecosystems that they target. These hybrids can provide unique genetic traits that promote survival in present-day environmental circumstances. And also, what is really interesting is this F2 hybrid. We have yet to find offspring of an F2 hybrid today. And I believe that they are actually out there. However, this hybrid taxa is not studied thoroughly throughout the Caribbean. And I'm hoping to encourage some of your students, some of you guys, to think about studying a crop of Prolifera, and hopefully more of us studying this hybrid, we can identify F2. And when we identify this, then we'll be able to answer a lot more interesting questions. One of the interesting phenomenons, if we are able to find an F2 hybrid, is something called reticulate evolution. Now, if you look at examples of this in the Indo-Pacific, which is a much older ocean, they've had millions and millions of more years of evolution. And in the Indo-Pacific, there are actually over 100 different species of acropora corals. And majority of them, upwards of 65 to 70%, are cross-fertile, so they can actually reproduce with one another. And we believe that the speciation, this explosion of diversity in terms of species, it was facilitated by reticulate evolution. Now, if we're able to find an F2 hybrid, and we're able to find subsequent generations of Prolifera colonies that are reproductively isolated from the parental species, so they can no longer backcross, then we will indeed have a brand new species in the Caribbean. And this would be one of the primary examples of reticulate evolution in the Caribbean, which I think is absolutely fascinating. I think that's so cool to think about, that you could potentially be witnessing evolution within our lifetimes. Another interesting paper, so I wish this paper was published back when I was still in grad school, but this paper, this seminal paper by Dr. Bill Pretsch, looked at the fossil records of certain locations in the Caribbean and identified that the hybridization is actually not a recent phenomenon. And thanks to the geological records, they've actually shown that there's evidence for high population density of all three of taxa. And so prior to the 1980s, there were also high densities of cervicornis, palmata, and Prolifera. Now, why is this missing from the history books? in this study, there are a lot of difficulties in distinguishing the difference between Prolifera and the parental species. And if you think back, it wasn't until the year 2000, thanks to the advancement of technology, that we were actually able to identify it was a hybrid due to molecular techniques. And so this history that we've been reading and learning about, the population decline of the Caribbean acropora species, most likely incorporated Prolifera. However, it was not as well covered in a lot of the research papers up until this point. And so with this understanding, provides another argument for why we want to incorporate Prolifera within coral restoration methodologies. Indeed, this hybrid taxa has an important ecological role in the Caribbean. Similar branching morphology to the parental species, occupies an intermediate niche, so it occupies a depth range in between both the parental species, provides a conduit, a continuing coverage of acropora corals that will provide all of those beneficial ecosystem services that I mentioned earlier. So future studies hopefully will look into the hybrid viability, will continue to look into the hybrid viability of acropora prolifera. Hopefully we'll be able to locate and identify F2 colonies and understand the physiological traits and how well this F2 generation can survive in present day ecological stressors. We will hopefully continue to do more genetic testing and more genetic studies to identify unique traits that allow this colonies to survive, like the intermediate branching morphology that might limit predation of certain types of predation. And with all of this continuing advancement in technology, we can continue to understand how to promote genetic diversity by using acropora prolifera within coral restoration methodologies. And so I wanted to thank you all for taking the time to listen to my talk today. I wanted to thank my great graduate committee for all their support and guidance and some of the funding that helped me with this, the NSF EPSCoR and Alana Vento grant at University of Virgin Islands. But I also wanted to extend a very huge thank you to Dr. Mateo Lopez Victoria and the Universidad de Javeriana from Cali for allowing me to talk with you guys today and allowing me to be here in Cali, Colombia to enjoy this beautiful, lush and tropical country of yours. I am really honored to be here and to enjoy this place. And with that, I will take any questions.
Thank you, Colin, for your presentation. Congratulations for your work. I will now open the room for questions, and we can do in two ways. I am sure you understand a couple of words in Spanish, so I will let the audience do their questions, whether in English or in Spanish. And if we have some troubles like getting the proper question, then I can translate. And I have now one here, which is just saying thank you. And if someone wants to do a direct question to Colin, now is the time. Si hay preguntas de los presentes, este es el momento para hacerlas.
I know I gave a lot of information. It's
Juan Manuel raised his hand, so go ahead, Juan. I think you can open your microphone, or Andrea, tienes que abrirle. Yeah, okay.
Well, good morning, Colin. Thank you very much for your presentation. My question was about the coral restoration methodologies. When you were talking about the coral tree method, I was wondering if that method could work with a massive coral species, because I see that the ramified species are like floating on the coral tree. So I was wondering if that would work with a species from a massive coral.
That is a great question. And yes, the answer is yes. There are actually several coral restoration groups who are focusing on the massive species. So the Orbicella, Flaviolata, Annularis, and Franksy. They're also looking at Montasteria cavernosa as well. And one of the most popular groups is called Moat Marine Lab, where they do what is called microfragmentation. So they'll take a massive colony and they'll fragment it to hundreds of fragments to 1 to 2 individual polyps, really tiny fragments. But what they've understood is that with these kinds of fragmentation, it stimulates growth in a rapid exponential way. And they have small pucks. They look like little pieces of dye, like a coin. And they will slide. They'll have a little foot that they'll slide into, and then they'll resuspend these into the water. So it kind of looks like tables full of these corals like that. And they'll have, I've worked with them in the wild, and we have to use a small toothbrush to clean in between to keep all the algae from growing. But they do use massive corals, massive bouldering corals within the trees, but then they also do them in land-based nurseries. And so a lot, even in the US Virgin Islands, for example, they have long wet tables filled with seawater that have current flow, and they have tables full of these small fragments that will allow to grow to a certain size, and then they'll actually reattach these fragments together to create maybe like an avocado-sized colony that they can actually then reestablish into the wilds.
Thank you,
Awesome,
Colin, for
Thank you
your
very much.
answer, and Juan for your question. Are there any more questions? ¿Alguien más tiene preguntas que quisiera hacerle a Colin? Yo voy a aprovechar mientras alguien más se anima. Colin, I want to ask you from a more applied perspective, thinking, for example, on Varadero, which is a reef you are about to get to know in person, but from which you already read a couple of papers. We have found Prolifera there. If you were asked to give some advice on starting with some pilot project of Prolifera restoration, what would be your suggestions?
That's a great question. So let me, I'm sure to follow up, I'm sure there are there's Cervicornis and Palmata is present there as well, right? Okay, yeah, so one of the first pilot studies is to look at the genetic diversity of the parental species to see how much genetic variation is present. And then if there is Prolifera, the suggestion would be is to incorporate all three within coral restoration methodologies. So incorporating all three, trying to target as many genetic, unique genotypes as possible, and primarily focusing on the parental species, and in a controlled, small manner, identifying unique, robust colonies of Prolifera that might show great response to thermal extremes or high growth rates, or that high number of apical tips might be another metric for saying, okay, these genets we can focus on, and incorporating them all within a close proximity together to hopefully continue to grow to a size where they can reproduce and where they can continue to exchange genetic information to try to increase genetic diversity. And it may be also looking at outplanting them within their niche. So Cervicornis, of course, focusing and outplanting them in the deeper locations of Varadero, and in Palmata in the shallow, and then Prolifera in between would also be useful.
Okay. Are there any more questions? Yes, Giancarlo, go ahead.
Hi, Colin. Do you hear me?
Yes.
Hi, thank you for your presentation.
My pleasure.
I have a question about how we can reduce the rate of predation. You say that in the tree nursery coral, the corals grow in a 360 manner. All the arms grow like like in a sphere. But then how we can reduce if we sow the corals on the bottom of the sea? For example, to omit or to avoid that the fireworm can eat the ramification of the coral?
That is a great question. It's actually kind of difficult to limit predation in certain locations just based on the density of predators that are already present. And so when thinking about where to outplant these corals, where do you want to place these corals into the substrate, there's a lot that goes into understanding what is the best place to do so. One of the first things that certain coral restoration teams will do is go into a site that they want to restore, and they will do ecological observations of present corals. How much of the corals that are already there are showing signs of predation? How many of these corals there are being inundated by algae or other things like, and what kinds of predation is there? Is it the Palmacanthus, the small damselfish that like to eat and create algal turfs on certain colonies? Is it the fireworms or the small snails? And so taking into consideration the populations of these predators, well, maybe you don't want to outplant there, and you'll try to choose a different location to outplant your corals. Now, let's say you don't have a choice. You only have so many of these areas, and you really need to outplant at these locations. Outplant a lot. Outplant a lot of different genets, a lot of different types of colonies. We have seen in the past this genetic variation, right? So some colonies have genotypes that respond better to certain stressors. And so incorporating a large group of different genetic groups, different colonies with unique genetic traits, may also benefit or increase the success of outplanting corals despite the number of predators present.
Okay, thank you. Yeah.
Thank you for the question.
That's the way here. Thank you.
Someone is writing me a question through WhatsApp because they are watching through YouTube, but weren't able to go inside the room in Zoom. Let's see if the questions arrives on time. Meanwhile, I don't know if someone else have a question from the audience here, from the people in the room right now. Well, the question is not arriving. Oh, yes, there it is. The question is, if experiments were performed by you or the people you know that might bring genes or genotypes from Prolifera back to the parentals species? That's the question from the student.
Experiments, okay, so there are lab conditions, lab settings, where they are actively crossing different genets, and genotypes of the cropper species. So during the spawning events, which is in August, and that's, if you guys ever go spawning, it's interesting, but it's a lot of work. It's like 2 weeks, every day you're in the water. You collect the gametes bundles, and you bring them back into the lab, and they try to actually cross multiple genets and trying to stimulate more increased genetic diversity. And so then, yes, to answer your question, you can. I don't know too many places that are doing that. There is Nova Southeastern University in the States is doing some work, Moat Marine Lab, FWC, Florida Fish and Wildlife Conservation Commission, which is a government-run organization that I used to work for, is doing similar stuff. And there's the Carmarby Institute in Curaçao, actually, with Seacorp International. They're doing some phenomenal work where instead of using a coral tree to propagate corals, they actually go out and collect the gametes during spawning. They bring them back into the lab, and they will cross multiple species, multiple genets. I don't know if they work with Prolifera yet, but they will cross Cervicornis and they'll cross and Palmata together. And they're trying to create a lot of genetic diversity, and they put them on these kind of tetrahedral shape, like blocks almost. And you'll see small fragments, you'll see individual recruits of these colonies, and they put them out into reef, and the shape of this structure locks into the reef and provides a base for these corals to grow. And so that is some work that's being done right now.
Okay, Colin, thank you very much. I think we ran out of time. I want to thank you again for giving us this interesting talk today, and also all the people that could join the talk through the Zoom room or through YouTube. And we hope we can have you back here in Cali in a year or so, telling us a little bit the research you are now performing with the corals and the history of some Varadero corals. Thank you very much, Colin, again.
Thank you.
A good day.
Yes, it was a pleasure meeting everybody. Thank you so much.
Bueno, un feliz día para todos y muchas gracias por estar aquí, Andrea, muchas gracias por el espacio, por el escenario. Feliz día para todos.
Thank you.
My pleasure, guys.
Ojalá.